So I am very happy to introduce Bob Datta. Is that right?
Yeah.
Bob, you drop your first name.
Yeah, my mother calls me Bobby. So I know that I'm not in trouble.
Okay. So, of course, you all saw the abstract, but let me nevertheless just spell out the trajectory that Bobby came here for, and that is his bachelor's degree in molecular biophysics, biochemistry, and history. Was that history of neuroscience, history of biology, just
intellectual history.
Oh, interesting. At Yale, and then a long MD, PhD, obviously, from Harvard Medical School, again in molecular neurobiology. Subsequently, he went to Columbia for 5 years postdoc, focusing on neural circuits. And after that, he arrived next door on Longwood Avenue in the Department of Neurobiology, where he now is associate professor in neurobiology at Harvard. So I heard, we haven't met, but I heard from several people sort of along the way, including Joel, saying that you've got to get Bob Datta for Action Club. And so I finally managed, and I'm happy to have so. And because then I actually looked up what you're actually doing, and there were a lot of key concepts that popped out, that is motion sequences, behavioral syllables, using machine learning to identify clusters. We often use the word primitives, but well, we can interchange this, what is syllables, units, etc. So I think there will be a lot of stimulating thoughts. The other thing to just mention, one hallmark of Bob Datta's research is the integration of sensation and action. How does sensation inform action at the neural level? How do these two seemingly distinct, or all too distinct in research, these two domains actually meet and do what ultimately leads to our very successful behavior? So with all that, I think there's a lot to hear and learn from you, but you need to get this mug.
Oh.
This is our...
Awesome. Thank you.
So that has
Excellent.
previous speakers on it.
Excellent.
And at some point when I do the next round, we'll be on it.
Excellent. Thank you so much. So I only have 41 slides, which for me more or less rounds to 0. And I know we have an hour and a half, and I'm told that you guys are going to interrupt a lot, which is good because I talk super fast and I mumble a lot. And so it probably isn't clear to you whether or not you don't understand what I'm saying versus I don't know what I'm talking about. And so we can clarify that if you ask me questions. So please feel free to interrupt. I'd be delighted to kind of clarify whatever I'm muddling. But I want to start with just a few slides that more or less explain, let's make sure one thing, that more or less just explain my perspective. So let's just get started. All right. So this guy is a deer mouse foraging for food in the wild. And this task depends on this animal's olfactory system. So if you go ahead and make neural recordings in this animal's olfactory cortex, it's easy, and this is like a thing that my lab does all the time, it's easy to kind of decode or identify representations for whatever the animal is smelling at any given moment in time. The olfactory cortex is part of a highly recurrent circuit. It's a lot like hippocampus. And so what that means is that through the right kind of lens, you can easily discern information about sense past. And there's definitely some predictive coding going on here. So if you look at the data in the right way, you can learn something about what the animal thinks it's going to smell in the near future. Now somehow this animal has to sort through all of this temporally admixed information, not just about smell, but from all of its senses, and alight on just the right action to express at any given moment in time to allow this animal to solve its current problem of existence, which is to find food. And unlike what is traditionally done in neuroscience labs, where you can kind of go through one of these sensory motor coupling cycles successfully and get a reward, in the real world, right, where it's tough, you have to generate hundreds or thousands of these little actions, behaviors, often in a prescribed sequence in order to reach your goal. And so this, kind of roughly speaking, is the broad problem that my lab works on. We're interested in how the brain organizes information about the sensory world that is often fluctuating on timescales of tens of milliseconds. And we're interested in how the brain takes advantage of that organized information to generate naturalistic patterns of behavior that are composed dynamically and on the fly in response to changing sensory cues. And as I'll show you, the behaviors that we're interested in are often organized at the sub-second time scale. Okay? So we're interested in this problem of sensory-motor coupling, and although I'm not going to talk about it at all today, we become increasingly interested in active sensing and in how an animal's overall pattern of movement inform its understanding of the world. But we'll skip that for today. So this problem of sensory-motor coupling obviously covers, I don't know, 80% of what the brain does. So it's not a really good research plan for anyone, including me. And so we've had to simplify this down, and we've decided to focus specifically on the sensory modality that is most important for most animals, and that is the sense of smell. Right? So in the wild, for most animals, ethologically relevant scents drive naturalistic patterns of behavior that allow animals to survive. And so we're interested in how information about scents is encoded and then decoded to generate behavior. It's a general framework. And today, I will spare you any discussion of sensation. We're going to more or less skip that, although you'll see, like, I'll sneak in a little bit of sensation here and there. But today, I'm just going to talk about action. So when my lab started, I had the following research plan. Here was the plan. I was going to take some mice. I was going to throw them into an arena. I was going to give them different odors. I was going to watch how their behavior changed. And then I would use like gene editing techniques and optogenetics to play with the brain and the genome. And Nature papers would ensue. And I'd get tenure, and then everything would be awesome. So that plan was a bad plan. And it was a bad plan largely because, if you look at an animal, in the simplest possible case, like here, this is a mouse, a C57 mouse, exploring an apparatus called a circular open field, which of course is just a bucket. Open field? Open. Yeah, it's just a bucket, right? You threw the animal in. Home Depot, right? Even in this very, very simple case, what you immediately appreciate is that you don't understand the animal's behavior at all, basically. If you let the animal just run about and do its thing, you don't understand what it's doing. And so if you just, but what, in contrast to the fact that we don't understand it, it's clear just from watching this animal that this animal's brain is generating a coherent pattern of behavior that's evolving in some sort of statistically predictable way over time. We just don't have access to any of that structure, right? And so now with modern machine learning techniques and machine vision techniques, you can do all sorts of things with this video, right? You can draw dots over the ears, the eyes, the nose. You can automatically track all the stuff about the mouse, but all those parameters that you measure are basically shadows of the thing you care about, which is kind of the underlying organization of this animal's time-varying behavior. Okay, so this is a problem that lots of people have thought about, and actually it's a problem which has been out of vogue for a long time. But it's worth talking about the people who thought about it, which of course are the ethologists, right? So the ethologists believed that all animal behavior, like this animal's behavior, is actually composed out of a series of behavioral modules, right? That the brain can place into a variety of sequences quite flexibly in order to compose a nearly infinite variety of actions, including this locomotory exploration here. And so a lot of what my lab has been interested in is in trying to falsify or explore this hypothesis, this ethological hypothesis of behavior, that animal behavior is built out of modules. And so we've been building systems that combine 3D machine vision technologies and data-driven unsupervised machine learning techniques to try to identify an underlying modular basis for rodent behavior. Reasoning that if we could do that, if we could kind of identify a set of modules that describe this animal's behavior, we'd not only be able to provide a much better description of the animal's behavior, which in the biomedical sciences is sorely needed. Right? This animal's just kind of described largely as moving. That's not useful. But also we'd be able to make predictions if we did this right. We'd be able to predict an animal's future behavioral state based upon its present. And if we really did a good job, we should be able to predict what's going on in this animal's brain as it generates behavior. Okay? We're good with that? Yeah.
How deep is that bucket?
It's deep enough that they can't jump out. So it's a 1.5 feet, 2 feet.
Really?
Yes.
So it's just perspective that makes it look like its nose is over the lip.
Yeah, exactly. It's just perspective. That's exactly right.
That's the bottom.
Yeah, it's the bottom of the bucket. Yeah, we're looking down at a mouse in a bucket. It's a bucket. Right? All right, it's the simplest possible thing we could think of, right? Okay, so we built the system.
That's always the tendency of staying on the rim.
Yeah, right, exactly. Yes. So we built the system, and we call it motion sequencing, or MoSeq for short. And because MoSeq has actually been published now for several years, I want to show you it rather than spend a lot of time describing it. I'm going to go through how it works pretty quickly, but you can stop me and we can talk about the details if you care, okay? Okay, so motion sequencing really has two parts. It has like a data part where you acquire a bunch of data, then it has an analytical part, and those parts are separate. In its current formulation, the data that we feed to motion sequencing, for the most part, although not exclusively, is 3D data describing the overall pose dynamics of the animal. And so we get those data by placing depth sensors or depth cameras above any apparatus we choose in the laboratory. And so what you're seeing heat mapped here is, again, of course, is a mouse in a bucket. But what you're seeing heat mapped here now at any given point is the animal's height. And so when the animal's head turns red, he's rearing or sniffing. Its back turns red, it's arched up towards the camera. And we've chosen specifically to focus on an animal's three-dimensional pose dynamics because they are encapsulating. If you understand the complete three-dimensional pose dynamics of any animal, you can infer every action they've generated, both with respect to themselves and with respect to the world. And so our perspective is that if we only had one data stream to work with, this is a pretty good one to pick. So we then take these 3D data and we feed these data to a computational model that instantiates this ethological hypothesis for behavior. That behavior is built out of stereotype modules that tumble one after the other somewhat predictably in time. So just briefly, for those of you who care about machine learning, I'll describe the innards of the model. So in this model, each behavioral module is described as an autoregressive process through high-dimensional pose space. And these modules are connected over time through the use of a semi-Markov model for temporal flexibility. And an important innovation in our model fitting procedures and our modeling is that we use a Bayesian framework. And so we use Bayesian nonparametric techniques to automatically identify the state number without any human supervision or intervention. And what that means in the net is that we feed this model these data and we run our fitting procedure, which is based upon statistical techniques like Gibbs sampling. And what motion sequencing spits out is effectively the number and the optimal set of behavioral syllables or modules that describes the behavior that's being generated by the mouse within any given experiment that we're feeding the model. And again,
The raw data, one more time. The raw data here.
Yes, this is raw data.
Just repeat that for me again.
Yeah, so the raw data here is 3D data.
3D of the mouse's
The mouse's body, yeah. So we're feeding essentially, there's no markers, there's no nothing. We're literally just feeding the pixels that describe the carapace of the animal. That's it. So obviously those pixels will reflect the underlying joint dynamics. None of that we're capturing directly. Everything's inferred.
So you take the center of gravity of that
Yeah.
patch? Or?
No, we take all of the pixels, all of them. So, and actually...
So you compute autoregressive model of each of the pixels?
Yes, so we take the pixels, we take all the pixels. Actually, there's a pre-processing step that I'm skipping here. We have an algorithm that automatically identifies the head and the tail of the animal, aligns the animal into a consensus space. We then do a little bit of dimensionality reduction, and the autoregressive process works on that space. Okay?
Still get extra autoregressive process.
Yeah. Yeah, exactly. And it actually works in the pixels. It's just that it's computationally very expensive, right? And who wants that?
And you won't
For
get
how
too
many
much.
pixels are then included?
That's a great question. Yeah, so this is very camera dependent, and as many of you know, these technologies are evolving very rapidly. And so in our current setups, I think the data that we feed the model are something like 120 by 120 pixels, something along that. So it's fairly high dimensional rich data.
You're probably getting like 30 hertz or something like that?
Yes, so in the data that I'm going to show you today, everything is running at 30 hertz, but we're switching to a 90 hertz system. And please remind me to talk, but that matters actually. And so please remind me to tell you about that and what it means, because I think it's very important, right? Because we're doing time series modeling here, right? So the time at which you acquire the data really matters. Actually, I'll just give it away the key point, which is that as you go faster, things could become more or less modular, right? Depending on whether or not God hated you or he liked you. And in this case, God liked me. So as you go faster, the boundaries that the model discovers between the modules becomes sharper, not blurrier. And that for us was an incredible relief, right? Because if it got blurrier, it would just mean that we're inventing something. But the fact that the boundaries get sharper means that modules might be a thing. Right?
Just to clear the confusion, is there any general...
You're confused already? I just started. This is slide 2.
I'm a little confused. Are we just saying that a general motion of that animal is at any time, these things, the patterns that you have below is like just how you will sequentially do with this?
I'm going to get to that. I'm only to this part of the slide. I haven't even gotten to this part yet. I'm only to this part of the slide. So basically, you take these pixels, you take these pixels that describe the mouse, give them to a model, and the model will discover the optimal number of modules. which we call, because we think they're components of body language, we call these syllables. Okay?
Syllable, but what I'm trying to understand, at any moment, can two of them be, it could be a linear combination of two?
No,
or is it
this
just
is
this
no.
or this
Yeah, this is not a basis set. So it is not a linear combination of anything. Yeah, these are all, so there's a couple of rules that are here that are like rules that are explicitly articulated by the model, and you can disagree with those rules, but those are the rules we went with. All animals must do a thing at a given moment in time. Every frame gets a label, and you only get one label. And that's,
It's like component rather, it's not a basis.
it's not a basis set. So we're not adding together things. We're
You have to make sure I get this. So you're doing some spatial processing first to reduce dimensionality just in space.
Yeah.
Just the-
Yeah, exactly. We should do a little PCA just to make it easier.
120 pixels is feeding a small number of
Yeah.
numbers to the model.
Yeah.
And you're basically segmenting it in time.
Yes.
With the idea that every interval is described by some autoregressive model.
Exactly.
You discover the order, you discover the boundaries.
Yeah.
Okay.
You have,
And so
There's some people here you should talk.
your semi-Markov, just to continue on this, your semi-Markov model
Yeah.
states are defined in terms of the AR coefficients? Okay.
Yes. All that is true. Everything that was just said is true.
Do you get pause? Like,
It's a great question. So in this implementation, we don't. We actually have. Do you get pause? You know, we're looking at it. We're looking from above, right? Right. So one of the things, right. So one of the things that's kind of good and bad here, right, is like, so why am I so enamored of this data? Actually, part of the reason is that I don't have to do any feature engineering. All the pixels are equally weighted. So once I start measuring paw position, I have to think about how to balance that in some rational way against the rest of the data.
You can only do one thing at a time, but clearly, if
Of course.
you add things like that, you'll get
Absolutely. You know, we have a formalism describing what we want to see, and that model captures, you know. But the interesting thing is that it turns out that it's not. These assumptions are not actually as unreasonable as you might imagine. You'll see.
And does the tail matter?
The tail. So the tail largely covariates. There's a lot of covariance in the data, and the tail largely is redundant. If you had only the tail, you could learn a lot about the mouse. But if you have the mouse, you don't really need the tail. Does it make any sense? Does that make any sense? Like, yeah.
Not to the mouse, but to the image.
Yeah, exactly. We do a lot of things like, you know, we are really concerned with how real this is. And so we do a lot of things like, if we include all the principal components and we get all the spatial data we possibly can, if we quadruple the spatial resolution, what do we get that's new? And our current thinking is that we don't get that much that's new, actually.
So as you increase the frame rate, the segment boundaries sharpen. At what frame rate do you no longer get significant improvements going higher?
Yeah, no, that's a really wonderful. I'm just going to wait because I'm talking a lot. I don't know. We've only gone up to I think a little over 100 hertz.
It's still getting better.
But marginally, yeah, it's pretty sharp. I mean, but marginally. I mean, at some point, behavior will look. There'll be some continuity. We definitely haven't gotten to that point, I think. These cameras are too slow. For example, there are many rodent behaviors that aren't well captured at 100 hertz even. So have you ever seen two mice fighting? Like they're like this ball, like this amazing ball that's probably at the millisecond scale. And so to capture, there are many dynamics we're definitely missing, right? And of course, we're again computing over the carapace. We're missing all the joints and all that stuff.
Just on the point of the sharpening of the segments, it's not about the continuity of successive segments,
Yeah.
but rather about the preciseness, the precision of the definition of these segments.
Yes, exactly. And that's a really important point. So the one thing I didn't tell you, which I will tell you now, is that there's essentially one prior we feed to the model. The prior is the timescale at which we think behavior is organized. Because you can imagine having a lens that you tune to, right? And it turns out, and I will defend this claim, that at least one timescale that's reasonable to consider is the sub-second timescale. And I will show you data that convinces us. But if you basically take these imaging data, which strangely had never really been explored before, and you just do simple signal processing, like a sub-second structure to the data emerges totally independently of any model. Like it just lives in the data structure. And that really gave us confidence that that's a reasonable kind of thing to think about. And I will show you evidence that's even more consistent with that as a claim, okay? So why don't we look at these syllables so you at least get a sense of what I'm talking about. So here on the left, again, is a 3D movie of a single mouse. And I'm gonna hit play. The mouse is gonna generate some behavior, and at some point a dot's gonna come over the animal. And when it does, the animal is gonna express a given behavioral syllable that I've looked at, and I post hoc named it. And I'm gonna call it the move it along syllable, okay? But this is a unit of behavior that MoSeq identified. I didn't tell it to look for this thing. The animal behaves, there's the dot, the animal kind of takes a few steps forward, it moves it along. Is this a coherent motif of action? Is it a stereotype thing, or is it just some blip? here are 30 separate examples of the same behavioral syllable drawn from 8 separate animals and 8 separate experiments. So I'm going to take all these individual movies, I'm going to overlay them all, I'm going to time lock them all to each other, to the center of when the syllable is getting expressed. So because these are different examples, when I hit play, you'll see all sorts of behaviors. But then when the dot comes over the animal, they all kind of coherently express this move it along syllable. And as this movie loops, what you'll see is, of course, there's some spatiotemporal diversity to the expression of each syllable. And that is entirely by design. Each syllable has associated with it a certain amount of spatiotemporal variance, which is modeled explicitly in a syllable-specific manner, out of acknowledgement that no behaviors are expressed the same exact way twice. So this is one behavioral syllable. Here are 6 more that are drawn from a typical experiment in the open field. In a typical experiment in the open field, MoSeq identifies something like 30 or 40 individual behavioral syllables, again, whose average duration is about 300 milliseconds. Although that duration distribution is quite wide, there's some whose mean is 100 milliseconds, there's some whose mean is closer to a second, and each of those duration distributions have different shapes. And you can see lots of different identifiable behaviors. You can see a bunch of behaviors you might not have thought to identify as an ethologist. You can see this behavior where the mice routinely try to escape the bucket, and they always fall back in, thank God. And so this is kind of a parts list for behavior that's automatically identified for us via motion sequencing. And you can, of course, think about how these parts are sequenced over time by just building a behavioral state map. So here in this representation, each node is a syllable whose diameter represents how often it's used. And the directional arrows represent the likelihood of transition from one behavioral syllable to the next.
What does the scrunch mean? The syllable which you said scrunch.
I think it's
when you go back.
Yeah.
Scrunch?
Yeah. What was they doing?
They're scrunching. They're scrunching.
Oh, the scrunching line. Okay.
Scrunch.
I don't know that.
It's a scrunch. Animal scrunching.
I don't know that.
That's what a scrunch is.
Do the syllables usually start with the animal stop and end with the animal stop?
No. There are many, many of them that are not like that, actually.
So are there then sort of linkers, coupling kind of
Yeah.
identify, or just
So that's-
mechanical transitions?
Yeah, that's
that are
right.
Necessary to
That's why we'd love to go to 1,000 hertz instead of 100, because I think then we begin to get access to that kind of question, right? There's computational challenges with that, but that's exactly what we'd want. We'd want to go faster in order to begin to learn about that. Our cameras just aren't fast enough to give us clarity on that issue, unfortunately. Okay, so what does the state map let us do? Of course, the cool thing it lets us do is predict the future, and I'm going to come back to that point because I think it's an important conceptual point. If we know the animal's in one state, we can predict what the animal's going to do accurately several seconds from now. But the other thing that you can do with these, of course, is encapsulate comprehensively, at least from the perspective of the model and the camera, everything the animal does in an experiment. And that's super important for biomedicine because we have no idea how to phenotype animals at all. And so we've successfully used this exact technique to kind of explicitly and objectively identify what happens when you mutate a gene, when you optogenetically tickle a circuit, when you give an animal drug. Any manipulation you can imagine, we have successfully used this kind of technique to reveal to us both expected and surprising changes in behavior. So from a kind of utilitarian perspective, this technique is super useful. Okay? And we found it to be useful.
So a garden variety mouse.
Yeah.
The map looks pretty much the same. Okay.
So this map is determined by the intersection of at least 2 things. One thing is the physical environment the animal is placed in. So if you just do a very simple experiment, take the mouse out of a bucket and you put it in a square, now the animal has a corner to interact with, and it gets there's a corner syllable. So that's good, but you want that to be true. And that's not found, that syllable is not discovered in the circle. But the other thing is that, of course, the genomic variation. So depending on the strain you're looking at, you'll get some significant differences in the deployment of the syllables, both in terms of their existence and in terms of the probabilities. And so we have phenotyped, a large number of standard inbred strains, and of the standard strains, kind of an average rule is that about 70% of the syllables are shared across the set, and about 30% of the syllables are unique to any given strain. One thing that we're doing quite actively is with my buddy Hopi Hoekstra, who's across the river. She's a mouse geneticist. She goes out into the wild and captures wild mouse strains, and then interbreeds them to F2, phenotypes them, and then sequences all their genomes to clone genes for behavioral variability. And we're literally using motion sequencing now on wild-derived Paramiscus strains to try to clone genes that control aspects of the syllables, the grammar, or both.
In speech recognition, typically
generate probability of a syllable rather than...
And that's exactly what happens here. It's a probabilistic method. And we take these approximate point estimates. We take these point estimates that are approximate.
Okay,
these point estimates to decide. Typically...
then you use sequences to impose the order.
Yeah, exactly. And typically, we do a lot of things that computer scientists might not do, but we as biologists do. So like we often will restart this model 1,000 times, take point estimates across the whole thing, try to get some certainty over what's going on, rather than just like saying, hey, it worked. Because for us, we actually, the ultimate goal for us is to relate this to what's going on in the animal's brain and its genome. And so we want to be as precise as we can, yeah. So we do a lot of those sorts of tests. And I think you might want, just to keep going with this, I think that you might want to ask that like a big fight that I had with my computational collaborators was like we should set aside half of the restarts and see how they match up and all that sort of, and so we've run all those controls. And it turns out there's so much structure in the data that as you restart this thing, you get the same answers every time. As you move the priors around, nothing happens. The thing seems to converge on a common solution because there's so much structure in the data per se.
Within a strain, do you see unique syllables that are unique to an individual?
I'm gonna tentatively answer that no. I could be wrong about that. I think I will answer a question I can't answer, if that's okay with you, which is, to what extent within a strain does the technique capture individual variability? And if you let me get 2-3 slides in and remind me to answer that question, we actually have mathematically dealt with this issue of individual variability. And the net is that motion sequencing is excellent at capturing individual variability around a mean. That's a really, really important point, because we've been talking about variability, and I think it's something we're really interested in. Okay, cool. All right, so I want to go back to this kind of idea about prediction, because I'm going to harp on a very particular point that maybe is interesting to you, maybe it's not, but it's important to us, which is that, in my world, there's this proliferation of techniques that let you explore the brain in great detail. Like we're having this renaissance. And we're just now beginning to kind of apply the same sorts of thinking that we've traditionally applied to brain to behavior, to try to link those two things together. What this means practically is that people are generating all sorts of methods and algorithms for describing animal behavior on an ongoing basis. And so a really exigent challenge we have as a field is coming up with some way of arbitrating amongst these methods. Like, how do you decide whether a method is good or bad, right? How do you decide that and this is the argument, these are arguments that ethologists have had with words for 100 years. My description of being, I can see 10 states, but you can see 8 states. So I just, I have better vision. These kinds of very semantic arguments. And so we've been thinking about this problem pretty hard, and there's 2 ways to think about it from our perspective. And we think that ultimately the solution to this is going to come from using some sort of predictive framework to choose between models. So this is the first, as far as I know, fully generative model for mouse behavior. And so one thing that you can imagine doing is building a bunch of different types of models and doing model selection by asking about held out performance over time. And so we've done a ton of that. So one thing you might ask is, well, maybe behavior is really continuous, it's not really segmented. So what happens if you build an AR model only? What happens if you dial in or out the hidden Markov component? So we've built all possible iterations of this, and it has convinced us. We have a standard, which is held out likelihoods, that allows us to know that it's important from the perspective of behavior to think of it as being modular. And in that sense, we've tested this ethological hypothesis, which is an interesting thing. It actually, as far as I know, has never been explicitly tested. Do you really get a better description of behavior if you describe it as being modular? Here, mathematically, we can show that answer is yes. So that's cool. And people will, in the future, right, because of deep learning, we're going to generate all sorts of generative models for behavior. That's just starting now, for mouse behavior I'm talking about. But that's going to happen. And we're looking forward to that, because then we can have a kind of dialogue where we use things like held outs as a way of thinking about whether a given answer is better or worse. But there's another kind of prediction you'd want to make, which is much more of a kind of a semantic and a practical prediction. And I want to talk about how we've been thinking about that. We need the second mode of prediction because not all ways of describing behavior, especially currently, are generative. So we have a lot of methods for describing behavior that are like, hey, let me take some behavioral data and cluster it using t-SNE. How does that match up with this? Is that better or worse? And how do you think about that? So we came up with the following strategy. So our strategy was to take 1,000 mice and to inject each mouse with one of 15 different neuro or psychoactive drugs at one of 3 to 8 doses. We throw them in the bucket we film them for 20, 30 minutes acutely, and now we have this very large data set that includes a ton of behavioral diversity. Then you can basically take each mouse and characterize it using whatever means you want, right? So in our case, in the example I will show you, although we've done many different things, we will compare motion sequencing to a traditional kind of scalar description of behavior, where you measure how fast the animal's moving and its angle of its head, and where it is in the bucket, all the things that like you get from a Nolde's ether vision system. You describe each mouse with those data, so you get this kind of laundry list of descriptions of the mice. You feed all the data to a simple linear classifier, and you just ask which behavioral description affords me better predictions over which drug and dose each animal received. Right? So it's an objective standard that allows you to compare across all methods. And for fun, I just want to show you some of the data because it's kind of fun to look at. So here, this is a subset of our data set. Here are some of the drugs we've tested, and here's just a couple of the doses that we've tested. And here are a subset of the data that we include in what we call a traditional behavioral fingerprint, the things that people have measured for 100 years. So we include the complete distributions of the animal's position in the bucket, how fast it was going, its lengths, and just because we want to handicap MoSeq effectively, we include the complete distribution of all of its heights, which is the thing that's not normally accessible with a 2D camera, but we have those data in the 3D camera off the sensor, so we include it. Okay? So this is kind of we can fingerprint each single mouse through scalars, or we can fingerprint it using motion sequencing. And so for scalars, actually, if you look at these data, it quickly becomes apparent why, for 100 years, people have described behavior like this. So if you look at high-dose haloperidol here, you see that the mouse's position is fixed in the arena and its speed is 0. And that is because high-dose haldol in mice does what it does in people, which is to knock them out. And you can see that just with your eyes in the data. That's really easy to understand. If you look at its opposite, like high-dose meth, you see that the position's at the edge, the speed is infinity, and that's because these mice are high as a kite and they're spinning around, just like people. And you can just see that in the data. You don't need any analytics for that. You get that, right? And so this representation of behavior actually makes a lot of sense. For motion sequencing, we decided just to include the 0th order data, which is how often each behavioral syllable is used. And we just make a little barcode out of this, and this looks like nonsense, as opposed to this, which is immediately interpretable. But if we feed these data to just a simple classifier, what you learn is that, and this is a confusion matrix, so on the x-axis is each drug that was the animal were treated with, and the y-axis is the actual performance of the classifier. Perfect performance is bright white along the diagonal. Traditional metrics are basically useless. 97% of the time, motion sequencing can tell you the name of the drug the animal was exposed to, and 93% of the time, it'll tell you the name of the drug and the dose. Right? It can separate out like even closely related doses of like an anxiolytic or something.
So what are these traditional metrics? Just very briefly.
Position, height, speed, ellipticity, angle of the head, heading, path tortuosity, all center entries, anything you can imagine, like when if you had an undergraduate and you're like, score these behavioral things, and you have this list, that entire list is part of the scalar summary.
Just from starting, did you try a more complicated or more sophisticated classifier than a linear classifier?
We could basically, it's not dependent on the classifier. So if you do a nonlinear classifier, you still, it's just a function of the data, basically. The other thing, though, you can do is you can generalize. So instead of just doing scalar stuff, you can begin to think about it in a more sophisticated way. So
Is this just measurement by measurement?
No, we did not do this measurement by measurement. We built this fingerprint. We can also do some feature engineering to try to maximize the scalar performance. That still never comes close to motion sequencing. But the other thing is that, this is a quantitative framework for comparing any description. And so, we've thought about this, and we decided to see whether we couldn't generalize this across different ways of thinking about behavior. So one basic objection to this comparison is that this is a beautiful time series model, and this is just a bunch of numbers in a bucket. So what happens if we feed these scalar data to motion sequencing? Well, so this is precision recall, and these are F1 scores. And what you learn from that is that you get better, and what that tells you is that time series modeling matters, but you don't do as well as MoSeq, and that tells you that 3D imaging matters. So this is like a super informative result, right? Like you've measured everything you can about the animal, and what it teaches you is that thinking about time is important, and that the pose dynamics themselves are also important. The other thing that we've done is to actually start to use this framework to head-to-head MoSeq against other kinds of methods. So in the mouse world, the best validated alternative method to MoSeq is actually a technique built by a friend of mine named Gordon Berman. It's a technique called Motion Mapper. And basically what you do is you take video data, you align it just the way that we align our data, then you run basically a wavelet transform over the data, and you take those transformed and dimensionally reduced data, and you feed them to t-SNE, and you do a bunch of water shedding. And you can cluster data together that way. And so and he's actually been he has a very impressive video where he does that to a human dancing the hokey pokey, and you get all the little steps of the hokey pokey, including the turn yourself around. It's impressive, actually. But if we go ahead and take our exact data and run them through this kind of clustering-based algorithm, again, you do worse than motion sequencing. And that tells you, again, that time series modeling is probably superior to kind of naive clustering, even with a sophisticated embedding. All right, so this, OK. Yeah.
When you give these animals drugs, do any new syllables will appear, or do the old syllables with different combinations and magnitudes come out?
That's a great question. So in this particular case, new syllables absolutely do appear. Like when you give an animal high-dose amphetamine, there are all these stereotypies that emerge that just aren't present in a normal mouse. So we co-train our model on all the data, and so what you see is that there's the presence of some syllables that are just zeroed out in other animals that are treated with other drugs. And I'm not going to show this data, but it turns out it's a very interesting feature of the data that turns out from a drug development perspective to be really useful. So you can imagine, right, that this classification performance could be supported by one of two schemes. So one scheme is that each behavioral syllable is like a key on a piano, and what each drug is doing is kind of like taking your arms and kind of pushing down on all of them just a little bit variably so you can tell everything apart. The other model is that maybe what drugs are playing chords. And if you're playing chords, you should be able to identify these kind of key syllables that are characteristic of each drug that allow that drug to be distinguished from all others. And it turns out that the latter thing is the case. Right? So for every drug, we can identify a set of 5 or 6, 7 syllables that are really characteristically changed by that drug. And the syllables are unique, they're characteristic of the drug. And what's important about that is if we do that same kind of experiment across genetic models of disease, like say autism models, there's plenty of those, what we typically find is the same thing, that like the gene is playing a chord of like 6, 7, 8 different syllables that are being changed. So what that means is that we can effectively intersect this chord and ask whether a candidate therapeutic is on target, off target, or both. And we can actually quantitate how on or off target a given drug is. So I'm not going to show you the data, but like the only one of 2, I think, drugs approved for human use in autism is this drug Risperdal or Risperidone. It's meant to kind of calm kids down and address their hyperactivity. And so if we take an autism model and we characterize it, we can identify a bunch of syllables that are associated with hyperactivity, and we can show that Risperdal actually doesn't fix any of those syllables. Instead, it just sedates the animal by turning down other normal movement syllables. Right? And it just basically proves that the drug is just a sedative. And it's all off target, right? And that's the thing that clinicians would be like, oh, of course. But like we can quantitate exactly how on or off target things are. So it kind of it allows us to articulate a strategy for drug development, essentially.
But in your analogy, does the drug add any new notes?
Yes. So depending on the drug, you might end up getting, the piano might end up getting bigger. So again, like there's syllables that are specific to the stereotypies. Or just to give you another example of that, the BTBR mouse, it exhibits these repetitive behaviors. Normal mice don't exhibit those behaviors. And so each individual tick the animal has is actually captured as a unique syllable. So we can use motion sequencing to count up how many different types of ticks does the BTBR animal have. Answer is 6. And we can count them all up. Like that's how many it has, because we have 6 syllables that correspond to those ticks.
So would you pick up repetitive syllables that are performed in a rhythmic or periodic sequence?
Yeah.
As a single syllable?
Yes. Yes. If the same behavior is repeated, it's a single syllable. And
loops on one syllable. On the graph, would it be self-loops?
Yes. Yeah. Exactly. So so I've excluded those from the graph as you see it, but there's lots of self transitions that are captured by the model.
And would that be the repetitive stuff?
Yeah. Exactly. Right. And sometimes there actually you can capture sequences of 3 syllables that compose the tick that are actually distinguishable behaviors, and you can just see the loop in the map. Yeah. Right. You see these nodes that just kind of they pop out at you. Right? They're very obvious in the data. And actually, those kinds of things are what motion sequencing is designed to do because it's data driven, right? So it's discovering structure where there's structure, and repetitive behavior is as structured as it gets, right? Okay, so I'm going to transition from kind of trying to convince you that this thing is useful to trying to explain how we're using it to understand the brain a little bit. So those experiments are inspired by the following kind of observation, which I think is an interesting one. So this is an odor box. So there are olfactometers at each one of these 4 corners. And what you're looking at here are, again, 30 separate experiments with 30 separate mice all overlaid. The average position of the mice across the entire experiment in gray in the background. And we're going to outline the body of each mouse here. And initially, we're just going to blow air through all of these ports. And so this is normal mouse open field behavior. They like the corners, they go around the edges, they will dart through the centers of the arena. This is just normal mouse behavior, no big deal. But now we're going to switch and blow a molecule called TMT through this corner. This is a molecule that's derived from fox feces that drives innate fear behaviors in the mouse. And so you see their behavioral state has completely changed. They now kind of huddle in this corner over here. They do this kind of approach and avoid behavior. There's a whole bunch of things that the odor is eliciting in the animal. And the question is, how does motion sequencing capture that change in behavioral state? Like what did it think happened to the animal's behavior? And so these are the two state maps associated with the control and the TMT exposed animals. And if you actually match up the syllables, what you find is that they're the exact same syllables in both cases. So despite the fact the animal's generating a bunch of new behaviors, those behaviors aren't being supported by new pose dynamics per se. Instead, you can see the grammar of behavior is dramatically different. And if you subtract these two maps off, you can see that. Here in blue are all the upregulated trajectories through that space, through the state space, and in red are all the downregulated trajectories. And so to our first approximation, every new thing the animal did that you saw the animal did when we gave the animal the odor is captured as a trajectory through the blue part of the map. And so now we can look at these trajectories objectively and ask, well, does that make any sense? And so now when I play these movies, when the dot comes over the animal, you're actually looking at a series of 3 blue syllables. And indeed, what you see are exactly those behaviors that we know are elicited by TMT, spatially organized freezing and spatially organized odor-evoked avoidance behaviors, right? So, and that was all automatically discovered by motion sequencing. We didn't, of course, tell it to look for any of the structure. It just comes out naturally in the analysis, right? And so what this means is actually something super duper important. It means that much as has been shown in worms and in flies, in mammals, new adaptive behaviors can be constructed by the brain by reshuffling the order of behavioral primitives, right? And so the question then is, where in the brain does that happen, right? What controls that process? How can we begin to think about that? And so that led us to think hard about the basal ganglia, and in particular, its input nucleus, the striatum. And I'm not going to give you a long lecture about the striatum, but I'm just going to remind you the key things about the striatum that are relevant to our discussion today. So the striatum is involved in many aspects of motor control. The two important things that we need to think about today are, one, the striatum has been directly implicated in the ongoing implementation of movements. And the second is that the striatum has been directly implicated in a process called action selection, the process through which an animal decides to do a thing at the expense of all other possible things. The striatum is composed largely of medium spiny neurons that are all inhibitory. These neurons come in one of 2 flavors. Half of them express a molecule called the dopamine D1 receptor. And these neurons make up the so-called direct pathway. And this circuit is thought to facilitate action, but no one really understands how it does that. The other half express the dopamine D2 receptor. And this is thought to interrupt or prevent or deselect actions. Although, again, no one really understands how that works. And no one really understands, certainly, how these 2 pathways interact to control naturalistic behaviors like a mouse running around in a bucket.
So when you say action selection, that could be, grammar doesn't have to be involved in that.
It doesn't, but it might.
It would,
yeah.
You could have a behavior who have the action selection.
Yes. Yeah, it could be true that grammar is a form of action selection or not. Right. Let's see. Okay. Right? Okay, right? Yeah.
You're going to say yes.
Otherwise, it'd be a weird talk. Yeah, yes. Okay, so the other thing you need to know is that Kent Berridge and John Fentress and colleagues in the 70s, 80s, and 90s did some incredibly painstaking work showing that neurons in the dorsolateral striatum specifically seem to encode information about naturalistic behavioral components and sequences, at least for rats as they express behaviors like grooming or play behaviors, okay? And so a postdoc in my lab was inspired by that observation and decided to ask whether or not the dorsolateral striatum in general encodes everything about behavioral components and sequences. So can we identify in that particular region of the brain explicit neural correlates for syllables, for grammar, or for both? The other thing he wanted to know was, what is the interaction between these 2 pathways as animals generate spontaneous, naturalistic, unrewarded behaviors? And so he built a special kind of system in which you can essentially perform something called photometry, which is the measurement of bulk neural activity in both of these pathways simultaneously. And so he basically put motion sequencing together with, as I'll show you, with electrophysiology and photometry and miniscope type recordings. And so essentially now we can perform motion sequencing in the context of ongoing interrogation of the nervous system using any modality, essentially. So if we put all these tools together, this is what we end up with. So this is a 3D movie of a mouse. Here we're going to digitally, this is actually how we perform the computation. We've digitally lined the mouse so its nose is to the right, its tail is to the left. This is a mouse exploring the circular open field. But you see this mouse is wearing a hat, and attached to the hat is a cable. And that's because we're performing photometry in the dorsolateral striatum, and we are, for the first time, simultaneously measuring activity in the direct and the indirect pathways all at once, okay? This movie is being compressed here into this movie. This movie is this movie, essentially. And if you just...
Wireless? Or do you have wireless?
For now, this is wired. So this is tethered. We will in the future go wireless. But this, yeah, it does modestly. And we can measure how much it affects it. Okay, so you can see this white movie. This is just these pixels, basically. Notice how striated these data are? Just by eye, this is raw data, right? And so this is where we get the prior, basically, that behavior is organized at the sub-second time scale. Because each of these little blocks, on average, is 300 milliseconds or so, on average, right? Underneath this is MoSeq capturing and identifying each behavioral syllable. And underneath this are the neural dynamics that are being expressed in the indirect and direct pathways. Question?
Have you ever imaged exercise using GPS?
We have never measured. We've done all sorts of recordings, but Beth Stevens has not convinced me to do astrocytes just yet.
So this animal is
She'll convince me soon.
Is this animal just running?
This animal is exploring this bucket freely. He's not just running. He's doing all sorts. Yeah, exactly. So actually, this is what we feed to the algorithm. It's kind of centered in the line data. But the animal's doing all sorts of things. It's rearing, it's stopping, it's pausing, it's grooming. It's doing the.
There are so many syllables everywhere.
Yeah, because the animal's doing all sorts of things. Any other questions? Okay, and the important thing here is that, like, if you look just from a neurobiological perspective, if you look at these data, you might imagine a model in which if D1 is go and D2 is stop, that these should always be anti-correlated, and you don't see that. You see that they're sometimes anti-correlated, but sometimes they're completely correlated. And so, like, kind of naive models about, direct meaning go and indirect meaning stop are not going to be correct because it's obviously not what we see in the data, right? But we do see some interesting things. So if we just kind of look at the data for a long time, and believe me, we just kind of stared at it for a long time, we notice that we often could observe as animals transition from one syllable to the next, a little flicker in one or both of the pathways. And if you just compute over the average, so on the x-axis here is time, with 0 being the time at which you switch from one syllable to the next, and on the y-axis, this is just neural activity. What you see is systematically, in the grand average, so this is, I don't know, hundreds of thousands or millions of syllable instances, you see a systematic flicker in activity in the dorsolateral striatum. So for us, this was incredibly important because it meant that motion sequencing is essentially identifying a kind of rhythmicity in behavior based on structure in the behavioral data. So now we stick this recording device into the striatum, and now we've discovered an exactly matching rhythm that had been previously undescribed in striatum. And the fact that this neural rhythm matches our behavioral rhythm is pretty suggestive that the segmentation that's being provided to us by MoSeq is reasonable.
Is there a delay between the two?
So there is a delay between the photometry and the behavior. And so to deal with that, we've performed simultaneous multi-unit electrophysiology and photometry. And that has taught us that if you just take the derivative of the signal, which is shown over here, you get a pretty good estimate of when actually firing occurs in the striatum. And that teaches you that the onset of this activity change is 50 milliseconds before syllable switching.
Can you just
But that's a good
explain
question.
the two panels here?
Sure.
First,
On the left.
They seem to more correlate.
Yeah, exactly. So this is the grand average. This is like you take 1 million syllables and the neural activity associated with them. You see this kind of global correlation. But that's if you average everything. Okay.
And then what's
And we'll talk about that. Yeah.
What's the what you're talking about?
This is just the derivative of this. And we plot, and I want to show this because this basically represents what's happening in the electrical activity underneath this optical signal. But
Since, don't you need like a control condition where you have transitions not between different syllables, but between something that is, just action related with any syllable or something like that?
So we have a different control because I think describing transitions here is challenging. The different control we have is just to stick these fibers somewhere else in the brain and to make sure that we don't see this kind of flickering everywhere, and we don't.
Yeah. I still think that, to say what is good, so it's going on here, something special, but is it transitions between syllables, I think,
I'm going to get to all that because there's a lot more information that we can obtain than just this transient. But yeah, but these are all reasonable questions. Like what this tells you is that there's something time locked between the brain and behavior, and that's all it tells you.
So the What
right?
you're saying is that intersyllable transitions.
Yeah, it seems like there's at the transition there seems to be a bump. But like, so the question you're let's just address the question you're asking, right? Which is, like, is this what's happening for every syllable? Or instead, maybe what's really happening is that there is some sort of unique amount of activity in each behavioral syllable that's associated, that like.
Or if you break it up in some way, if you synchronize it with something that's not syllables, but something else that's action related, that's common across syllables.
Yeah. Okay. So we've done that as well. So basically, if you try to regress any of this neural data against any scalar metric that you can imagine, you get nothing.
Yeah, okay.
You get nothing. So you don't get speed, you don't get acceleration, you don't get any of that. You don't get height, you don't get length. Any of that traditional metrics thing, you basically don't get any of it. Correlations are like 0.1. So there's a little bit of something, there's a little bit of something, but not a lot. And that's of course that's a main thing, right? Like, it's like, are syllables really a thing, or are we really measuring something else? And so we have done that. Okay, so is this a uniform feature of all syllables, or instead maybe is each syllable associated with a unique amount of activity in each of these pathways? So to address that, we can just color code each syllable here. It's just arbitrarily color coded. And we can just plot out on the x-axis activity in the direct pathway, and on the y-axis activity in the indirect pathway. And so here we're averaging across what we see in like 12 animals. So again, many, many, many thousands of instances in each case. And this is just a single animal example. And what you see is that indeed, each behavioral syllable is associated with a unique amount of activity in the direct and the indirect pathways. And that suggests that there's a kind of neural code in which a certain amount of activity in each one of these pathways corresponds to the expression of a particular set of three-dimensional pose dynamics.
Regardless of the transition.
Regardless of the transition. I think that's kind of just worth noting just for your head, is that if you average here as well, like we were talking about, the pathways are correlated, here things look pretty correlated. So we do a bunch of averaging, things look pretty correlated. Okay? But there are some syllables where things are a little bit decorrelated.
It's interesting that to be clustered would be one thing, but to have them be correlated like that is
Right.
Extra.
Right. So yeah. So there's a theory. So there's this kind of stop and go theory. That's kind of a theory of folks like, and these are cartoon theories. They're not real theories. But like Anatole Kreitzer would kind of articulate a model that, in the broad sense, maybe there's a stop and go thing that's going on. Other folks like Rui Costa would actually predict that these pathways should be simultaneously active as part of the process through which action selection is refined. And so like they would so like this kind of conveniently enough is evidence for both models. Right. Okay, so these, of course, are averages, and what I'm hiding in these averages are all the neural dynamics that are associated with each syllable. Let's just pick 3 syllables and look at that. So here are 3 syllables, and again, we're averaging over 10,000 or so instances of each syllable. In red is the direct pathway, and green is the indirect pathway. And even though we're doing this ton of averaging, you see that each individual behavioral syllable I've chosen here is associated with a unique pattern of activity in the direct and the indirect pathways. And furthermore, if you have a proper time alignment and you're not just kind of globally averaging everything, now all of a sudden you see fairly sharp decorrelations in the data at fine time scales, right? 100 millisecond, 200 millisecond time scales. Okay, so the fact that each one of these behavioral syllables was associated with a kind of unique pattern of neural activity led us to ask whether or not there was a systematic relationship between behavior on the one hand and neural activity on the other.
So let me say something heretical. Isn't that non-surprising that there is a correlation between the neural activity and behavior? But what made you look at the D1, D2 rather than, well, usually motor cortex and things like that?
This is part of a longer conversation.
Okay.
I think this is not surprising. It's surprising in some senses and not surprising in others. So, and I'll just kind of cut to the chase of my argument, which is, I started this talk by telling you that in cortex, and olfactory cortex, the past, present, and the future are all there. That's true in motor cortex, of course, as well. But I'm not certain how true it is in striatum. What's amazing here to me is that there is a rhythm that's defining for the mouse, and I'll convince you that this rhythm is real, as kind of I showed the last few slides, that essentially is crystallizing and defining the mouse's behavioral present, right? And so we have a very broad model for corticostriatal function in which the purpose of these interactions between cortex and striatum is actually to distill time down to the present, so the animal has some sense of what it's doing in the moment. And that I would not have predicted. That's the model for us, the interesting model that emerges from the data, and that's not something I would have naively predicted. Okay.
Curious also, but so we keep emphasizing you're averaging a lot of instances here, and we can't really see from this picture what the tails look like on those. Like, how can is this really stereotypical behavior, or is it? You know what I'm saying?
Yes, I'm actually not going to.
Is this something that's kind of deterministic behavior, not anything but stochastic? Or?
I will directly address your question with math.
Okay.
Is that okay?
That's what you could do.
Exactly. All right. So I will. But let me say another thing first. Okay. So like the fact that we have these associations makes you wonder whether or not the relationship between behavior and neurobiology is systematic. That is, do similar syllables, are they associated with similar patterns of neural activity? So in order to get a handle on that, you have to somehow embed these behavioral syllables in a space that lets you understand nearest neighbor relationships. And of course, these syllables are autoregressive processes, so that's not the simplest thing in the world to do. But my collaborators are excellent, and we're able to actually generate an embedding, not the best embedding, but a workable embedding for these behavioral syllables to let us at least understand, nearest neighbor and local relationships amongst these behavioral syllables, which let us kind of objectively identify, similar behavioral syllables. They did some time-dilated thing and then measured some distance and some space, I think. They played the autoregressive processes forward. They synthesized a bunch of things. They time-dilated them, and they looked at some form of distance metric. I don't know which one they used. It's some form. It's not great. It worked for this purpose.
There are, especially speech techniques for
Right, so there's a
distances
distinction.
between objects.
That they would use LBC, like AR
You use
models.
capture. There are all these techniques of developments.
Yeah, it's funny. So the person who actually did it is a birdsong person, but I don't think that he actually did any of those things. I would love to talk to you more about that. That's an important problem for us moving forward for lots of reasons. So if you want to, I'd love to discuss that more with you. Okay, but we have this kind of workable embedding, and so we can pull.
First row, you claim that is similar?
Yes, I do claim that is similar. I do. But different. Right, they're different from each other, right, but they're roughly similar. These are different from each other, but they're roughly similar. And that's what you'd expect, right? They're different syllables, so they should be different, but they're not dissimilar from each other. And so if you kind of globally look across the data at all the correlations, what you find are if you just consider the red trace or just the green trace, you get pretty weakish correlations between neural activity, neural distances and behavioral distances. But if you consider them both and you concatenate them, you do much better, actually. And so there is this kind of global correlated structure. I'm not certain I would have expected that, actually. From the birdsong literature, to be honest, like in the birdsong, that's not what's observed. So in the birdsong, right, you have completely distinct neurons that are encoding these behaviors independent of the relationship. And so, right, it's completely different. And so like this systematic encoding of behavioral space, 3D behavioral space in the striatum is a thing you'd want in your brain, but I'm not certain I would have predicted that it would live here in this form. Okay, so again, these are averages, right? And I've been complaining about that this whole time. But we can do another thing. We can do the thing that you really care about, which is to instead of implanting a fibrophotometry device, we can instead implant a camera and image cellular level activity in individual spiny projection neurons as we perform motion sequencing. And then that allows us to extract traces of individual neurons in the direct pathway or in the indirect pathway. And now we can look at these ensembles of neurons and ask very basic questions about, at the ensemble level, now no longer averaging, at the single cell level, what information lives in this space? Okay, so the first thing, you'll appreciate that the first thing I then did with these data was to average it, just as a reality check. So if you just average those data, you get exactly the main result that I showed you before, which is that as you switch from syllable to syllable, on average, this is global fluctuation. You can also do exactly the kind of analysis I showed you before, which is to identify ensembles and ask whether related ensembles correlate with the expression of related behaviors. And that turns out to be true. But the thing you really care about is, are individual neural ensembles specifically associated with individual syllables? Right? That's basically the question. And so we have individual neurons, and so we can build a decoder. And it turns out that if you ask a decoder to identify specifically which of 41 separate behavioral states the animal is expressing, 25% of the time, based on the neural data, I can tell you exactly which syllable it is. And if I loosen that criteria to do a syllable or its nearest neighbor, it's right about 70% of the time. So there's a huge amount of information in the striatum. Yeah, right? Yeah, there's a huge amount of information that lives in the striatum that specifies the identity of the ongoing syllable. This is I should never have included this in this way. This is a way too complicated panel, but I will tell you with words that you can also explicitly decode the sequence context in which a given syllable is expressed. So it turns out, I'm sorry, I'll just describe with my hands. So it turns out, in both the photometry data and in the single cell data, imagine you have a syllable B. So B can be embedded in a sequence A, B, C, and imagine that's a high probability sequence. Or B can be embedded in D, B, E, and that's a low probability sequence. And we can show, because motion sequencing doesn't just give us the autoregressive process, we actually know the actual kinematics as the animal generates the syllable. So we can show that B is actually kinematically identical in both of these cases. And if we compare the neural representation for B in a high probability and a low probability context, we find that during high probability sequences, there's much less neural activity. And during low probability sequences, there's much more neural activity. And so 90% of the time, I can tell you whether a given syllable is being embedded as part of a highly probable or highly improbable sequence just by looking at how active the ensemble is within a given syllable. So that means that there's kind of explicit neural information about both syllables and sequencing in striatum all at once. And that definitely wouldn't have been predicted. That's like,
Now, wildly interpreting and extrapolating here, does it mean that the striatum then is responsible for stringing things together,
Okay.
as opposed to then once it is strung together, it is controlled elsewhere?
Let's ask that question. So maybe it's stringing things together. So that's one model, because we have this kind of sequence data. Maybe it's required for implementation. That's the thing the striatum does. So maybe it's required for implementing the syllables. Maybe it's both. Maybe it's neither. So my lab did the easiest possible experiment, which is to bilaterally lesion the dorsolateral striatum. Okay, and then in these lesioned animals, you take the structure out, and then you just ask how is behavior changed using motion sequencing, which we can do with high fidelity. And so here's what you see. This is a control animal. This is a lesion animal. And by eye, these animals look pretty healthy, these lesion animals, right? Looks to you like it's normal behavior. This is 3D imaging. If we motion sequence these animals, the syllables that are expressed in this animal are identical to the syllables that are expressed in this animal, okay? And they're morphologically identical. And so that rules out a model in which the dorsolateral striatum is strictly required for the implementation of these syllables, despite the fact that there's like complete decodability of the syllables in the striatum, which is amazing, right? There's a code that's not being used for implementation. Okay, but if we now plot out the behavioral state map of these syllables, you see it's dramatically changed. So here in the sham case, we're going to put all the syllables on the outside of the ring so you can see the transitions really clearly. And you see that in the normal case, the behavioral state map is relatively low entropy. There's a lot of rules that govern the sequencing between these different behavioral syllables. And you see that those rules are dissolved, and behavior becomes randomized if you lesion the dorsolateral striatum. Okay, so this proves that the DLS is required for the appropriate sequencing of naturalistic behaviors. The problem with this claim is I just showed you a movie of this animal looking completely normal. So this is just like some quantitative result that I can be all proud of, but it means nothing potentially. So how do we think about that? Well, I'll remind you I showed you this I showed you what happens if you give TMT to an animal. What happens is that animal's behavior completely changes, and it changes entirely due to a grammatical change. So what happens if I take the sham animal and this lesion animal, and I give these animals TMT? So on the left is the sham case, and TMT is just going to be blown through a port. And you'll see that the mouse will go, it'll approach the TMT, it'll smell it, it thinks there's a fox there, and it runs away. And I don't know, have any of you worked with mice? But they're idiots. And so this mouse is going to go back to the TMT, right, because he's a moron, and then he's going to run away again, right?
Just wants to make sure.
Yeah, exactly. Just want to make sure it's not going to die, right? Yeah, very smart animal. If you look in the lesion case, what you find is that the mouse, even though we know it's sensing TMT, behaves like the TMT isn't there at all. It's completely incapable of producing de novo a sequence that allows it to generate an adaptive behavior. And this really demonstrates that the role of the dorsolateral striatum in stringing together behavioral components is behaviorally meaningful, because the animal can no longer avoid being eaten in this particular case. Okay? So I just want to end by mentioning 3 technical things that we're doing. We're doing many, many things to improve motion sequencing. There's 3 technical things that we're doing that I'll just kind of mention briefly. These are just really brief anecdotes, just to give you a sense of kind of where our head is. So one thing is that, of course, all I've done so far is shown you mice in buckets. Mice in buckets are boring. You can see this is a mouse in a box, very boring. But if you put 2 mice in a box, all of a sudden things get very, very exciting. They have all these rich interactions. And one of the problems is that because we're computing in pixel space, it's hard to disambiguate these mice for motion sequencing. But I have a super smart guy in my lab who's beginning to be able to do that through some deep learning techniques. And so that lets us, for the first time, be able to actually take a couple mice and do some motion sequencing over them as they freely interact. And so imagine you have 2 Balb C mice. this is the simplest possible experiment. You just take 2 bulb sea mice that are individually housed and you put them together. What do you see? So what you see is immediately when you put them together, immediately their syllable usage starts to separate, with one mouse expressing one pattern of syllable usage. So the syllable, which syllable is being expressed is on this axis, how often it's used in the experiments on this axis. This is just a simple usage plot. Right? You see there's like a separation in the usage of these particular behavioral syllables. And over time, as we kind of familiarize the animals with each other trial on trial, we see that these exact syllable differences become wider and wider until the animals crystallize into a classic dominant submissive relationship. And that's what these syllable differences are actually encoding. So what's neat here is, of course, you can begin to think about the ontogeny of dominance relationships and submissive relationships, and that's interesting. You might think about using this kind of technique to predict, based upon an animal's behavior by itself, its solitary behavior, can you predict whether it'll be dominant or submissive? But those are secondary questions to us. The exciting question to us is about the joint structure in the data. So now, for the first time, we have now 2 behavioral data streams and 2 sets of syllables. And so we should be able to use math to objectively know when these 2 animals are behaviorally coupled and when these 2 animals are behaviorally decoupled and they're behaving as if they're kind of in a solitary context. And that's important because it means that in principle, we should be able to know when 2 animals are socially coupled at a distance. And basically the way we measure social coupling in mouse models and rat models is when they're touching. Like that's how you score social behavior in all of these disease models. When in truth, we all know that we all have body language that's real and we use it to communicate. It's certainly true for rodents as well. And we can now potentially capture those kind of social interactions objectively by having a behavioral measure of social coupling. And so we're super excited about that as a possibility here. The second thing is that, of course, we're interested in kind of testing mechanistic hypotheses about how different kinds of neural circuits, corticostriatal circuits, other circuits, are involved in generating individual syllables or sequences. Okay? And so if we want to attack that as a problem, we need some method in which, as an animal is freely behaving, we can on the fly identify what syllable he's expressing, and then through optogenetics, tweak their brain in some way. And so in order to do that, we've built a CNN-based framework that allows us, with 30-second millisecond latencies, to identify individual syllables with very high accuracy. And so armed with this kind of ability to perform closed-loop motion sequencing, we're now implanting fibers all over the animal's brain and seeing what happens as we manipulate neural circuits. I'll show you a very simple toy experiment in which we afford the animal virtual dopamine every time the animal expresses a particular syllable. And what happens is, after just 4 or 5 stimulations, the animal's usage of the targeted syllable goes way up. And to me, that's a little bit of an expected result. It's amazing that it works, but it's a little bit of an expected result. There are many questions you want to ask about that, like how does that affect adjacent syllables? How does it affect syllables that are part of different sequences? We're working on those things. But to me, the really interesting thing is that if we do this experiment, this reinforcement doesn't bleed over into the most closely related syllable. And that doesn't prove anything, but it suggests on the surface that maybe the brain actually is discretizing syllables in a way that it can identify that we're reinforcing this thing and not this thing. So we think that's pretty interesting, and clearly as a general framework, this is going to be very powerful. And the last thing I want to mention is that, of course, we describe behavior at the sub-second time scale with good reason, but we all know that behavior is at many interdigitated time scales organized. And so we're finally now building hierarchical models that kind of learn a much longer time scale structure of behavior. And our hope is that through the use of these hierarchical models, we'll be able to do things like apply motion sequencing to long-term monitoring of animals, to do things like potentially monitor a mouse across its lifetime to understand the ontogeny of behavior and how its behavior is hierarchically organized at many time scales. And that's another thing we're working on. And with that, I'll just mention I'll remind you that we've kind of come up with this motion sequencing thing, and the things that we're really interested in now are trying to understand how sensory information arrives in DLS to influence sequencing. We're interested in actually mechanistically how the dorsolateral striatum selects actions and strings them together into sequences. And of course, this thing that I haven't talked about is how do those action sequences affect sensation in turn? And so we're really in the lab thinking about all 3 of these problems now. And with that, I'll just mention that most of the work that I've talked to you was done by a spectacular postdoc of mine named Jeff Markowitz. He's an expert song guy, along with a student of mine named Wynn Gillis. There's a computational fellow in my lab named Caleb Weinrab, who's now working on the social stuff. And Diego is working on the hierarchical stuff. And all this work is done as part of a collaboration with my neighbor and friend, Bernardo Sabatini. Thank you again for your attention.
Maybe as you are, sorry, just one thing. As you mentioned to me, you also have some human work just to
Yeah.
sort of
Yeah, yeah, it's- and I'll-
see
yeah,
how,
yeah, sure.
how the methods can
That's
be
right.
used in.
And so, this general modeling framework, these autoregressive processes with switching, like they capture all sorts of features of behavioral data really nicely, right? This looks pretty good. So accelerometer data looks great with our model. You know what's naturally syllabic or syllables, like when you talk? And so the ARHMM is working beautifully over facial gesture data now. And so we have a bunch of collaborations with folks. So for example, we're collaborating with Justin Baker at Mass General. He has accelerometers on all the patients in the bipolar ward. And we're trying to use the ARHMM to predict, the motion sequencing to predict whether patients will have outbursts, for example, which is like an important medical thing for them. We've been taking advantage of some data sets that have been made available to us to try to do pain prediction based upon facial gestures, and that actually is going pretty well. There's some ground truth that's associated with those facial gestures, and we can pretty easily decode how painful a person thinks a stimulus is. That kind of affords the possibility of getting past the 1 to 10, smiley face, frowny face scale that we have in the hospitals. And so we think this is a pretty generally applicable framework. An important thing here is, as I mentioned before, the analytical side and the data side are really separated. And so we've been dialing in and out all sorts of different data types. So the thing here we've been doing, for example, is ultrasonic vocalizations, right? Like animals, as they socially interact, they sing to each other. That has a ton of temporal structure. So we've built a thing called VoSeq, which is motion sequencing for vocalizations. No one's ever actually built a generative model for mouse vocalizations, and this is really useful. So we've been doing things in collaboration with my friend Lisa Stowers, where we play back the generative model to actual mice to see whether they're fooled into thinking a conspecific is there. That's an important way to validate the model, right? Currently, all people are doing are clustering. Like, is that representation of behavior good or bad? A way you can decide is if the mouse thinks it's good or bad, right? So we do this generative thing. So we think this is a very flexible framework, and we're using it both in humans and in mice in various ways.
Question about that. Do you see anything qualitatively different in humans, or are humans just big rats?
That is a I can't answer that question, actually. We don't have the data to really answer that question. So for facial gesture recognition, which is the thing we have the most data on, things are not that qualitatively different. I mean, it really looks syllabic. It looks, if you do the same kind of just dimensionality reduction over the raw video data after you've embedded it somehow, it looks blocky. So it seems pretty fundamentally, at least that seems pretty fundamentally similar. I would want to talk more to you about the accelerometer data, but the data is so low dimensional inherently that it's hard to draw general conclusions. So I don't want to overinterpret those data. Mm-hmm.
So a very exciting talk. Couple of questions actually. So in syllables, in speech, right? You have this phenomenon called articulation, which is that one syllable is affected by the preceding and following syllables. If I heard you correctly, you said that there was no such effect in the behaviors that you observe here.
Right. So here, just so I can understand exactly what you're saying, what you mean is that the kinematics of a given syllable should be dependent on the context in which. So I'm not saying that that's not true, because our model has a lot of variance in it. I'm saying that that could be true, but the way that we're looking at the data now, I wouldn't know it. Okay. So that's possible.
Similarly, related to
Just
this.
on that, I want to hear
Just on that, the co-articulations usually of phonemes or gestures within syllables.
I see.
And so if you have your effect on syllables by context is kind of co-articulation.
I see, yeah, okay.
But in co-articulation at the phony level can go beyond syllables, those are rare.
Okay.
Maybe it's like fairly local and its activations overlapping.
I see.
But
I see.
I interpreted the syllables as phonemes here because there's no difference between, it just sounds better.
Yes, exactly. That's why we use the term.
So
it's all made up anyway, right?
So the co-articulations are adjacent to the,
yeah.
Okay. But it is a...
Peter Perona calls these movemes. Right?
Yeah.
So you're not finding a grammar yet?
In the sense of a formal grammar, no.
No, okay.
Yeah.
And so I was wondering, since you're observing this foraging behavior essentially, or something like that, right? Which actually is characterized by random walk almost, right? Sort of random superposition synchronization or concatenation of these syllables, right? I was wondering if you give them
think so.
a little bit more of objective.
Yeah,
like
yes,
for
yes.
example, that Fox now.
Yeah, okay.
Yes.
Yes, okay, great. So I actually have a different idea about this,
Okay.
and I'll tell you what my idea is, and I want to work on this idea, and you can tell me that I'm stupid. Here's the idea. So here's a problem I want to work on. I want to understand, okay, so like I have a black painted empty bucket, and I throw a mouse in there. Why does the mouse do anything at all?
Right.
And then, after like surprising me by doing stuff, why does it stop doing stuff? Right? So that's what happens, right? Like it explores for 20, 30 minutes, 15 minutes, and then all of a sudden it stops. Right? Yeah, and so it's obvious what's happening. What's happening is that the animal has a model of the world. You violate that model, even though there's nothing in the environment. And so what the animal is doing is generating these exploratory behaviors. So in this kind of, if you frame it like that, then each behavioral syllable is a saccade and has some information value. And what the animal is doing is trying to kind of asymptote to some level at which its predictions about the world match its perceptions, right? And it's acquiring information as it generates these behaviors. And then once it's done that, it's like, I'm out. I can just sit around because my models now are perfect. And so what that means is that in principle, if I could figure out a way to do it, we should be able to build an arena where we can dynamically manipulate sensory information and measure the information value of each movement. And that would let us model, this kind of basic exploration behavior. It's never been done, right? It's super interesting. I think we had the atomization of behavior gives us the tools to do it. So like I would love to do the experiment. I would love to do the experiment.
Something like introduce the smell.
Yes, exactly. Yeah, exactly.
Turn the system.
That's exactly.
Individual.
That's right. One of the challenges here, one of the basic challenges here, is that as a general matter, sensory information is poorly parameterized. So like what makes two sensory stimuli similar or different, and how do you think about that? So the other half of my lab actually, just for totally different reasons, works a lot on that problem, like how similar or different are two different odorants. But we have ways of actually measuring that now, and so we think that will allow us to do this kind of experiment in the long run.
Just a philosophical point, my personal philosophy
Right.
is that while I totally agree with the information gathering through action, I think
Infotaxis.
there is also action just for action's sake.
Yeah.
Because a lot of the modeling always has this idea of humans are lazy. What is the minimum effort? What is the
That's right.
minimum?
That's right.
Humans die if you make them sit in a corner
Yeah.
for days. So just
It is,
wild
yeah,
idea.
it is an assumption that they're doing something optimal, and they definitely could be violating that assumption. I
But
agree.
it could be even in that totally boring environment.
Yeah.
At some point, they just need to exercise, right?
Yeah.
So that there is another spontaneous set
Okay, so
that is not for information gathering.
Yes.
Just for getting rid of
So
energy.
that's actually why we want to do this hierarchical modeling, because you would imagine that exploration and exercising look different if you look at a longer timescale view. So we really want to look at non-stationarities to give us a sense of, so that lets us, in principle, infer something about the underlying internal state of the animal, right? Which is kind of what you're getting at, like what are its internal motivations? So wouldn't that be awesome? I'm not certain it's true, but like one can imagine a model in which if you had a perfect description of behavior, you could actually infer all the internal states. Maybe not. There's many reasons to think that you would not be able to do that, but it's possible that you could. So that's the thing that we're really thinking about, exactly this.
So I think in a way kind of related question. I have a colleague here who would probably shoot me if I didn't ask something about this. So, her argument would be that a lot of the brain's interest is in maintaining the internal body state.
Yeah.
Which you kind of ignoring your original.
Yes, I'm definitely ignoring, yeah.
So is there some way to kind of incorporate that into what you're doing? It's kind of related to producing a move because you need to self-regulate by moving, perhaps.
Right. So I think so one of the, okay, so let's step back for a moment and I'll tell you my strategy for thinking about this. So why am I excited about the two social thing? Like I'm excited because that's interesting, but I'm also excited because it's the first step towards taking a covariate and asking if we can jointly model behavior in some covariate. So that will allow us in the long run, when we get dense neural recordings, to jointly infer neurobiology and behavior, which is of course the right way to do this in the long run, not through regression. And then of course you can do things like measure every neuromodulator in the brain and ask whether or not you can't regress the overall structure of behavior against that. And the things you're talking about, like internal state, those will be reflected in those neuromodulators. Yeah. So it's of course a super important question. When we do this mouse in a bucket thing, part of why we do it is because it's very stressful for the animal, and essentially we homogenize the animal's behavioral state by like freaking him out by throwing him in this unusual bucket. But things will get much.
See the same thing you do for movement, right?
I'm sorry?
Do you see the same kinds of same primitives, the same syllables in free movement?
Yeah.
Right, so
Yeah, exactly.
You're not just,
Yeah.
you're not only finding out what mice do in their body.
Yeah, that's right. That's right.
So is the question about co-articulation was kind of leading in a direction that I was thinking about. So how small is a syllable or a unit, and at what point can they not be discrete
Yeah.
any longer? So at some point the muscles have to do it, so there's going to be overlap and there's going to be co-articulation. So is there some limit to the technique, to the time scale, at which switches between syllables that are unchunked into bigger syllables? So what are the elemental units, I guess?
Yeah.
Is there a way to get down to that?
Yeah.
Or is it you're working on this time scale and up to lifetime, which is
Yeah,
great.
but can you get faster?
Yeah.
Yeah, so as I mentioned, it's clear that even from observing mice by eye, they do behaviors that are much faster than this. It is not clear that we have the ability to subfractionate these syllables into smaller time bins in a way that makes sense.
Yeah, I mean, this is.
But that does not mean that that's not true. Like, it just means that just given our modeling framework, there's nothing in the data that suggests that we should be able to do that. It's possible that with much faster cameras, it would be obvious that we should do that. Right? We just don't know.
The question I asked at the very beginning about tailless mice
Yeah.
was an interest in balance, which
Yeah.
can be looked at as a continuous.
Exactly. Exactly.
Decision,
Okay.
It's often looked at as this simple, feedback.
Yes.
model
So
kind of thing,
let's talk. You're getting it right. You're getting it like an essential thing that we haven't talked about. Okay. So there are many, many limits to what's being done here. One of the limits is that we're computing over the carapace of the animal. So what we're looking at is like this meat sack, right? Basically, that's like reflecting the things that you care about, but aren't actually the things that you care about, right? What you care about are the joint dynamics and the muscles. We're missing all of that. So one idea that we have is, which would kind of improve the modeling and actually bring our work more into the framework of the sorts of things that you think about, is to try to do a little bit better machine vision with multiple cameras and to build a true, complete 3D model of the mouse that includes the paws. And then, and not that we care about the paws per se, but what we do care about is the joints. So then we can potentially learn a joint model from these 3D data, and that would allow us to now reformulate the model, not as an ARHMM, but as a dynamical systems, as a set of a switching linear dynamical systems model, which we've been thinking about, where there's a kind of, we're just within each syllable are a bunch of joint dynamics, and they switch states, and that will probably much better reflect what the brain cares about, because that's what the brain cares about. The brain doesn't care about the meat sack; it cares about the joint dynamics, right?
So why do you have to switch models? Why I guess I'm wondering why joint angles can't be like pixels.
We just have to measure them. We have to somehow measure those joint angles, and we're missing them in our current model.
Yeah.
Given that, we need to get those data.
Have you ever done motion seek on mice while they're sleeping? And could you use your generative model to understand what's going on when there is no motion, but there's a lot of brain activity?
So the answer to the latter question is definitely no. No. Yeah. So we're actually doing a bunch of that stuff. We've been collaborating with Yvan Soltesz. So he's an epilepsy guy who's been using MoSeq for longer-term monitoring in the context of field recordings to build better Racine scales to look at what happens through sleep. And that all seems like it's working pretty nicely. So you can use it to discover different stages. And again, actually, what's interesting there is, how many stages of sleep are there really? Right? How many, right? Like during epilepsy, are there really 6 stages or not? And so like one of the things this is going to give us the ability to do is to get maybe a little bit more of an objective sense of how external and internal state are linked and how many states there are.
Yeah, it maybe would be cool to look at the D1, D2 activation during sleep, after one of your sessions, to see if you could guess what was happening in the absence of motion.
Yeah, exactly. Can you? You're asking like, is there a version of replay that we can capture about the animal's 3-dimensional kinematics as opposed to what people do in the hippocampus, which is to capture just their path through space? I was just actually out with Lauren Frank at UCSF. We're talking about just that problem. Yeah.
One confusion and a question. So the confusion question first, that when you put these 2 mice together and you check their syllables, so you use the word dominant, and I was not sure what you meant by that.
Yeah, so what I mean is, if you put 2 mice male mice that have been singly housed inevitably when they come together, they fight a ton, and there's a winner and a loser. And the winner, and then like if you put them together subsequently, basically like the submissive mouse is like this, and actually he has some interesting syllables that like are pretty unusual. So one thing that they do actually is they kind of huddle in the corner, but they're always doing like this, like to check out whether they're going to get beaten up. You know, it's stuff like that. It's really interesting actually.
So in that case, then you also see the hope that in some other pair of mice, there could be, you could figure out if one mice is, for example, there could be a teacher-student relationship.
Yeah, all that, yeah, exactly, all that sort of thing, yeah.
Okay. And the other one is, if you make a histogram, you talked about like a particular syllable happening between two other neighboring syllables and the distribution of that. How long can you make these like sequence of syllables, like words, sentences, which still pop up?
Yeah, that's
Do you see sentences?
you know, honestly, that's an amazing question. We just haven't looked at the neural data carefully enough to know the answer. That's a really wonderful. So I think, like, so what did I not show you? Like, what is the thing that I like skipped right over? Like, I didn't model any of the neural data and then show you that the model for neural data maps onto behavior. I only modeled the behavior and showed that it maps onto the neural. Exactly. And so obviously we're working on that. That's a little bit challenging in the sense that firing in the striatum is pretty sparse. So like those neurons don't fire that often, and not that many of them fire, and the firing doesn't last for the whole syllable, right? The kind of, and even on a single trial, it doesn't tile the syllable, at least, you know, for the several hundred neurons that we can image. And so it's not like a nice cortical code where you have like ongoing activity. So one thing we're doing is we're taking advantage of these new neural pixels probes to simultaneously record in motor cortex, dorsolateral striatum, and substantia nigra. And I think from those recordings, I might be able to answer your question.
But even without the neuronal part, if you just do it even in the motion sequence, it tells you if there is like some fewer number of goals.
Yeah, exactly. Yeah, we haven't done that. It's a really good suggestion. It's this thing we haven't approached yet.
It's sort of the same, same question, less specific.
Mhm.
Can you, your current identification, are there distinct timescales? Meaning, what is the average one for a syllable? And if you have loops, sequences, is there another timescale emerging?
Yes, that's a great question. So, you know, again, these syllables have a mean duration, but they're each characterized by a duration distribution, right? Which is, you know, defined by the semi-Markov process. So it's between exponential and Gaussian. So some are long, some are short, right? And they have their own little shapes. If you take the syllables and you spectrally cluster them, it's clear that there's neighborhood structure in this graph. And if you look and if you explore carefully, it's clear that there are longer timescales that you can empirically identify from the data. So one timescale we've been playing with that we know exists naturalistic in the data is about the 2 to 3 second timescale, which kind of comports with your intuition about mouse behavior, right? So it's like they express 6 to 12 syllables and they kind of pause and reevaluate, maybe hop to another part of the map. That's
Is there anything clock-like about the time scales, that the transitions, you know, can only occur on certain ticks or?
right. So our optogenetic experiments are beginning to allow us to do that. So you might imagine that like an experiment we're definitely doing is like optogenetically intervening in the striatum per se in the middle of a syllable and asking, can I interrupt it? Right. Another thing we're doing that we're super excited about.
I mean, those are really tough experiments because outside the striatum, you can interrupt, you know, the Adam Hantman work, you know, you can interrupt, you know, instantaneously. You know,
Adam has reminded me of that. so there's a converse thing that we can do, which I think is kind of similar, which gets a somewhat oblique thing, right? Which is, you know, I've been claiming that there's this rhythmicity. So can we manipulate the overall time scale? So one thing we're doing is just in training the animal with dopamine pulses that are super brief, but like 100 millisecond, 200 millisecond, 500 millisecond. Can we move around the global time scale? It's fun, right? Like that's the thing we can try. So that's the thing we're doing.
So if you were to do something like take an action like walking, right, and then you hurt the animal, maybe you like break its leg or something like that, or constrain it in some way, would you see something like the same neural activity, right? Even though the behavior had, like the output behavior had
That's a
changed,
great question.
Maybe the intention is the same. Have you looked at anything like that?
Yeah, so in the part of the brain that we're looking at, it's almost certainly, these neural representations for the behavior themselves seem almost certain to be an admixture of commands that are coming down from motor cortex and a reflection of the ongoing behavior from sensory information that's filtering in. So I would absolutely anticipate that if I broke the animal's legs, these codes would change. That's for sure. An interesting question that relates to this, though, is, you know, if this is really a code, then the code shouldn't be context specific, right? So is the association between neural activity and a given syllable, does that change when I give the animal TMT? And that answer is no. So even though the whole pattern of behavior is different, the syllables still have the same code. And that's like just a gut check. That's like, that's actually a code, right?
So on the same thought, would you say that these syllables then are general, so even if you have the minds out of the bucket,
Yeah.
you will still find the same?
Oh yeah. I mean, the syllables are shaped by context. What I'm doing when I'm speaking is different from what I do at home, right? So they're shaped, but given an appropriate context, the syllables are the same. Yeah. So just an explicit example, elevated plus maze versus a bucket. You know, most of the syllables are overlapping because most of what they do in a bucket is to run around in a line, and that's what they do in an elevated plus maze. But the things that are different, like stretch attends, that only happen in the elevated plus maze, they don't happen in the bucket.
So in the very beginning, you mentioned the reason why you studied to this kind of work instead of traditional way of
Yeah.
working, when studying on olfactory system.
That's right.
Yeah, and I'm just not clear, how much you feel comfortable with this, or what actually makes you feel better, instead of doing what you meant to do,
Mhm.
what's the real difference between the traditional method and what you're doing, at a more higher level, like in terms of scientific question or the approach.
I see what you're asking. So yeah, so basically so imagine how I would study the response to TMT. I would measure how often the animal froze. That's the standard metric. And it's allocentric position in the arena. Those are incredibly noisy metrics that only they don't tell you actually anything about how the brain generated that behavior. By atomizing behavior, I could show that it's grammatical. I could identify a part of the brain that commands it. And I understand, and now it prompts a key scientific question, which is, how does sensory information filter into a structure that generates grammar? None of that would have been accessible to me if I just measured freezing. Right.
I would have been surprised if they hadn't been.
I think that Megan's question, so you did something that at first time the changes movement patterns.
Yeah.
How do these things adapt at all? Do they just compose their change in action from existing syllables with a different structure, or? I think that's what you were asking.
Yeah,
right?
exactly.
Is this adapting?
We've never done an experiment where the animal is forced to suddenly change its overall, where we teleport the animal effectively from one context to another. We've never done that. Yeah. We have done experiments that don't quite get at your question, where we optogenetically tickle motor cortex and force the animal to generate strange behaviors that it normally doesn't generate. And we can watch how that perturbs the global structure of the normal behavior of the animal. And and what happens is and
Natural response to something.
yeah, that's actually and what that shows you is just kind of how I would say resilient the global structure of behavior actually is, because when you do that, actually animals quickly just kind of fall back into the normal state map. But we haven't done exactly the experiment you're asking about, which is a good experiment.
Just have to add a little ankle weight.
Yeah, exactly. Let's pour molasses into the arena, see how they do, right?
It
Well, I mean-
sounds like your technology sort of antiquates classical fear conditioning scoring systems.
Yeah.
I'm wondering how hard it would be for a new researcher to implement your cameras and algorithms. Is there an out-of-the-box system that you
Right.
have made available?
So I have two answers to that. One is I'm collaborating with Becca Shansky here to actually adapt this to fear conditioning.
Yeah.
So that's being done. And the second is that this is not out of the box at all. It's freely available, but it's a huge lift. I got a U-24 grant from the NIH to hire a Google engineer to productize this for academic distribution for free. Right, and so the first milestone is a beta version that everyone can have with a GUI by the end of the year. So either you get a version in a year or I lose the money. So hopefully you'll get your version in a year.
It'll be a lot better than Roll and Rock.
I'm really excited. I mean, I'm really excited about distributing this technology. It's just a heavy lift.
You had one more?
Yeah,
was it?
no, I have two more. First, earlier I asked you a question on syllables unique to the individual. Did you say everything you wanted to about that?
I didn't. Now I can. Thank you for bringing it back up. Okay, so I showed you that you get much better discriminative performance with MoSeq versus traditional metrics, and the fact that we see that allows us to ask the question why. So we can actually go into the data and say why does one work better than the other in terms of classification. two possible answers, right? One possible answer is that we have 10, 12 animals, 20 animals per condition. And so maybe in motion sequencing, the variance collapses, or maybe the means are better separated, or some combination of the two. So when we look at the data and we compare motion sequencing to conventional metrics, what we find is that the variance is actually much bigger. But the means are much better separated. And so we get much better classification performance, but this individual variance that we see is much bigger. And if we subsample the data, that individual variance is biological, it's not technical. And that tells us that motion sequencing is capturing individual variability. But we needed this drug experiment to know that. And that kind of prompts the possibility that we can study individual variability using the technique. So we're excited about that.
And my second question was, when I last talked to you about this research was 3 or 4 years ago, and you said you were getting a lot of pushback from the field.
Yes.
I wondered, is it any less now?
Yes.
And why is that? What are their complaints about this?
So there was 2 complaints at the beginning. So one is that this is trivial. So the complaint number 1 is we basically understand behavior. It's a thing that the animal does, and measuring more precisely is stupid. So you're stupid. So that's the first set of complaints. And the second set of complaints was much more sophisticated, but no less damning, which was this is a trivial model, and you're imposing structure on the data, even though we've done a million controls to show that that's not true. And so you've generated some approximate thing, and so that's stupid too, so you're stupid. So those are the 2 main complaints. One is it's impossible, and one of it's already solved. So those are the 2 things. Right. So I think, right, yeah. It's solved or it's impossible. So it was my favorite set of reviews ever. That was amazing, right? And I think a couple of things have happened in the intervening time. So one is I think the importance of this as a question has really grown as our access to neural data has really improved. It's just obvious that this is a problem now. It's a real problem. So you might have seen these recent papers from Nick Steinmetz and Churchland, and Matteo Carandini, where they have dense probes or they're imaging somewhere in the brain through some means in a head-fixed animal. And they have high-density video recordings of the animal. And most of the animal's neural variability is just the animal fidgeting. No one ever even measures it. But most of what's going on is the animal twitches a little bit for whatever reason. So I think people are just generally accepting now that this is a problem. I think it's taken me a long time, just to be honest about the process, it's taken me a long time to come up with language that is evocative enough for people that they can grasp what we're talking about here, which is this process, for birdsong people, they've had words for this for a long time. These ideas are less common, so this had to be some language improvement. And I think the neural data is now convincing. Now you can actually show that you can make predictions. And that means that this is not a completely arbitrary description of behavior. And I think that helps people grab onto it and believe that it's a thing. Even if it's not a perfect thing, it's at least a thing.
I've just been thinking of classical ballet, which is sort of the archetypal.
Yeah. Laba notation, right? How do you?
Yes,
right?
it's archetypal that there is a limited set of movements
That's right.
that are practiced
That's right.
over and over again, and then put into these
complex sequences, exactly.
And so, in terms of testing, probably your algorithms on human data, you could look at a classical sequence.
I would love
and
that.
then identify, basically coming back to the bar exercises that they're doing, versus, of course, then another dance type which doesn't have that.
Yeah.
So while it probably and hopefully will just say exactly that, it is a verification in a different context.
Validation here is really challenging. How do you think about this as being, you can, I think actually what's interesting here conceptually is that since the development of behavior, since the invention by Sturtevant, right, or whoever, of behavioral neurogenetics, the standard that we apply to whether a behavioral description is worthwhile is whether it's useful. So even if it's totally craptastic, if you can clone a gene, it's good enough, right? Even if it's horrible, whatever, forced swim, if you can find a circuit, it's good enough. And so we don't have to think about it very hard because utility is the standard, but we're definitely moving to a place where that's no longer going to be true, where we're going to have to think more carefully about what makes a better or worse segmentation. So validation becomes a real challenge. And so things like, does a thing generalize across different data sets? I think this is incredibly valuable. Again, various forms of prediction I think are going to be incredibly valuable. And this is not a problem that people have actually thought about very seriously for behavior, right? This is a thing we're inventing as we go.
Okay.
Great, thank you so much.